drug synthesis and mechanism of action. So in this unit, in today's class, I am going to cover basic concepts of mechanism of action, introduction to macromolecular targets like proteins, carbohydrates, nucleic acids, as well as lipids, right? And classification of drugs, enzyme inhibition and its types. So what are the types of enzyme inhibition are there? So these are the topics I am going to cover in this class. So hopefully you will learn many basic things about drug synthesis and mechanism of action. First of all, let me define you about drug. Drug is a chemical substance which interacts with the macromolecular targets in the body and produces biological or pharmacological effects. So that is called drug. It may be synthesized or it may be artificial one, right? So whichever is useful to treat the diseases, they can be considered as drugs, right? You know about synthesis. Being a chemist, you know about synthesis, chemical synthesis. So we can synthesize the required compound during some steps, so by applying some methodologies, by taking some starting materials, reagents, right, some catalysts, right. These are the way we can synthesize some important bioactive molecules. They may be act as a drug, right? So first of all, let me define you about mechanism of action. What is the mechanism of action of a drug? The specific biochemical interaction through which a drug substance produces its pharmacological effects. So that is called mechanism of action of a drug. Okay, it is nothing but a specific biochemical interaction. So there is an interaction between a drug and the macromolecular target in the body. So with that interaction, it alters the pharmacological effects or the physiological effects in the body. Okay, so that is mechanism of action. So mechanism of action is nothing but the specific biochemical interaction between a drug and macromolecular target in the body. Okay, our mechanism of action is nothing but a study of drug effects. So a specific term is there for this, that is pharmacodynamics. Pharmacodynamics is nothing but it can study the drug effect in the body. Okay, so I'll give you one example. Suppose you take aspirin. Okay, aspirin, the mechanism of action of aspirin involves irreversible inhibition of the enzyme called cyclooxygenase. That is one of the enzyme. Aspirin, a drug which goes into the body and interact with an enzyme called cyclooxygenase with irreversible inhibition. Inhibition is stopping the action of that enzyme, cyclooxygenase. Therefore, suppressing the production of prostaglandins and thromboxanes, thus reducing pain and inflammation. Okay, so that is the pharmacological effect with the aspirin, right? So aspirin goes into our body and it interacts with the cyclooxygenase which is an enzyme, and with the mechanism of irreversible inhibition, and stops the production of prostaglandins and thromboxanes. So finally, the pharmacological effect is reducing the pain and inflammation. So that's what happens when aspirin takes as a drug. So that is the kind of mechanism of action of aspirin. So some principles of drug action alter the pace of ongoing activity, not impart the new function. Okay, the main principle of drug action, it alters the phase of ongoing activity but not impart the new function. Okay, so that is the basic principle of drug action. There are some types of drug actions like stimulation, depression, irritation, replacement, cytotoxic action, antimicrobial action, modification of immune system or immune status. Okay, these are the types of drug actions. It may be stimulation. By stimulation, it acts. By depression, stimulation means enhancement of the activity, particular activity. Depression means reduce the or decrease the particular activity in the body. Irritation, by irritation also drug acts and gives some useful benefits to the mankind. Suppose if you take some Jendu balm or Amrutanshan, so if you apply that on your head, it gives some irritation, some kind of different sense, but it reduces the headache, sometimes body pains, and for cold, it is a naturally many people uses like that, right? Replacement, by replacing particular uh, things, it acts in the body. And cytotoxic action means the drug goes into the body and into the cell, and it destroy or it kills that infected cells or infected cells, right? And also antimicrobial action. Some drugs will show that the kind of drugs will enter into the body and it fight against the microorganism and it kills that microorganisms by interacting with them. Okay, modification of immune status. By this way also drug acts in the body, right? That means immune status. These days many people are talking and discussing about immune system because of coronavirus, right? So coronavirus, the best thing to face the coronavirus is to develop or improve the immune system. So that's what everybody is suggesting, right? So if you change the immune system status, you can also, that kind of drugs also useful, beneficial to the mankind. Okay, so these are the types of drug actions. It may be stimulation, it may be depression, it may be irritation, it may be replacement, it may be cytotoxic action, antimicrobial action, or modification of immune status, right? So these are the basic things we need to understand about mechanism of action of a drug. Okay, if you want to understand the mechanism of action of a drug, so you need to know the definition. The definition is I already explained, right? It is a specific biochemical interaction between drug and macromolecular targets. So finally it produces pharmacological effect or physiological effect beneficial to the patient. So this kind of study also can be called as pharmacodynamics. Okay, so I explained about aspirin, the mechanism of action of aspirin. Aspirin goes into the body, okay, and it interacts with a specific enzyme, cyclooxygenase, with irreversible inhibition, therefore suppressing the production of prostaglandins and thromboxanes. So finally it reduces the pain and inflammation. Okay, so these are the basic things you need to understand to understand about the mechanism of action of a drug, right? So let me explain you about interaction of macromolecular targets. What is macromolecular target? Macromolecule is a large molecule, very large molecule, such as protein, commonly composed of polymerization of a smaller subunits called amino acids. They are typically composed of thousands of atoms or more. Okay, so that's why we can call them macromolecular targets, right? Macromolecules means very large molecule. They have high molecular weights. They have some thousands of atoms which can connected by covalently. Example, protein. In protein there are some thousands of chains of amino acids, right? The and the most common macromolecules are proteins, as I said, nucleic acids, carbohydrates and lipids. Okay, so these are the four major type of macromolecules. So you are going to learn about these macromolecular targets. These are the very very important macromolecular targets. Among these, proteins are the largest group. The largest group, most of the drugs can target proteins. Most of the successful drugs which are available in the market, they targets proteins mainly. So therefore it is very important to understand the structure of protein and basic things about proteins. And also nucleic acids, you know about nucleic acids, DNA and RNA. Deoxyribonucleic acids, ribonucleic acids, they are also very very important part of cells. So they have genetical code. They instruct the they are like genes. They produce the information and they copy the information. They can communicate the information. They can synthesize the proteins, right? And also carbohydrates, you know, very very important organic molecules, and also lipids, of course. So these are the four macromolecular targets a drug can target, these macromolecular targets. So first of all, let me introduce you, or let me explain you about proteins. Okay, as I said, majority of drugs used in medicine are targeted on proteins. Proteins such as receptors and enzymes transport proteins. These are the three things, three kinds of proteins which are very very important. So many drugs can target these things. Okay, receptors, enzymes and transport proteins, right? So these are these three molecules comes under proteins, right? They are very large, okay, and very important proteins, which plays important role in the cell cycle systems and all the biological systems, right? So therefore it is important to understand the protein structure in order to understand the drug action on proteins. So if you're not able to understand the structure of proteins and all those things, it is very difficult to understand the mechanism of action or the drug action on proteins. Proteins have four level of structures. Proteins have four level of structures: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure, the individual amino acids making up the protein are linked through the peptide bond. Linked through the peptide bonds. So that is the primary structure of protein. So if you clarify the complexity of proteins, so finally you will get amino acids as a monomer units, right? Some thousands of amino acids linked together form a polypeptide chain through the peptide bonds, and that simple structure can be considered as primary structure of protein. Okay, so I'll give you two examples about one of the smallest protein molecule is insulin, right? And the largest protein molecule, titin, which consists of 34,350 amino acids in its structure. How many amino acids linked in that structure? 34,350 amino acids exist in titin, a protein molecule. Okay, so that is the largest protein which consists more number of amino acids in its structure. As I said, primary structure of proteins, as all the amino acids linked together sequentially and form a polypeptide chain. So if you want to understand about protein molecules, you need to know about amino acids. Okay, generally the common amino acids, if you remember 20 amino acids, so most of the protein structures consist these kind of 20 to 25 amino acids repeatedly. So the different proteins have different sequence of amino acids. The only difference in between different proteins are enzymes, receptors, transport proteins, all these things. The only difference is the sequence of amino acids. Okay, therefore because of the difference in their sequence, so they have different functions. So therefore the only thing, the sequence of amino acids are different in different proteins. Suppose one enzyme has different kind of sequence of amino acids, another enzyme has different kind of sequence amino acids. So those two enzymes, different enzymes functions are different. They have different functions. Okay, so therefore, so if you familiar with 20 or 25 amino acids, then it is very easy to understand proteins structures, primary structure and secondary structure, tertiary structure and quaternary structure. Okay, suppose amino acids can be classified into non-polar and polar amino acids, and positive charge amino acids and negative charge amino acids. The basic amino acid, glycine, right? So all these amino acids, they have only different R group. Okay, so in all the amino acids, they have amine group at one side, carboxylic group other side. In the middle carbon, the substitution of R is different for all the amino acids. That substitution, if you remember different substitutions, say for example glycine, the R is equal to CH3, no, not CH3, H. Okay, the R is equal to H. In the case of alanine, the R is equal to CH3. So I already so many times remembered about amino acids in the class, and I also sometime I remembered I asked you to remember all those 20 amino acids names as well as structures. You should be familiar to write the 20 amino acid structures. Then it will help you a lot to understand about the structures. Okay, so what about secondary structure? In primary structure, amino acids come together, link together through peptide bonds and form a polypeptide chain. Okay, and at one end it has amine group, at the other hand it has carboxylic group. So that is about primary structure of protein. Simple. Okay, sequence of amino acids form a polypeptide, okay, through peptide bonds. That is the primary structure of protein. So what about secondary structure of protein? So if it is forms polypeptides, the large chains, okay, so there is a chance of hydrogen bonding in between. Okay, in secondary structure you have two kinds of forms, protein chain forms, polypeptide chain forms. One is alpha helix and another one is beta fleeted sheet. Okay, so these two are protein secondary structure. So what is the difference between primary structure and secondary structure? In primary structure you have only peptide bonds in between amino acids, that's why the chain grows. In the secondary structure of protein you have hydrogen bonding in between. Okay, that large chain, it may coil like this, like alpha helix. So in between chains, the C double bond O and NH groups, that hydrogen and that oxygen, so they forms hydrogen bond in between. Okay, so that's how it forms alpha helix, and sometimes it forms beta pleated sheets, like open book, or if you fold the paper and if you open it, so you will see some folds, so that is called beta fleeted sheet. Okay, so in beta fleeted sheet, so there is a another hydrogen bonding between the two chains. So one polypeptide chain and another polypeptide chain in between, so there is a hydrogen bonding. Okay, so in alpha helix only that chain, it goes like this, coil like this. In beta pleated sheet, so if you fold a paper into so many folds and if you open it, so you will get that beta pleated structure. In beta pleated structure there is also hydrogen bonding in between one chain to another chain, right? So that's how two kinds of alpha helix and beta pleated sheets will be formed. So those two are called secondary structure of proteins. So what about tertiary structure? Tertiary structure occurs when certain attractions are present between alpha helices and beta fleeted sheets, right? Tertiary structure occurs when alpha helix and beta pleated sheets, when come together, there is some attraction in between them, so they stay together. alpha helix and beta pleated sheets. They will form some kinds of large chain. Okay. Tertiary protein structure occurs when certain attractions are present between alpha helix and beta pleated sheets. Okay, that is about tertiary structure. In primary structure, only amino acid chain. In secondary structures, they folded like alpha helix and beta pleated sheet, and in between there are hydrogen bonding. And in tertiary structure, some interactions, some attractions occurs between the alpha helix as well as beta pleated sheets. So they come together, they stay together because of those interactions. That's how they form tertiary structure of proteins. So finally, quaternary protein structure is a protein consisting of more than one amino acid chain, more than one polypeptides. Okay, quaternary structure is the final structure. You can see when more than one polypeptide chains come together and they form quaternary structure of protein. So these are the four kinds of protein structure are there. Okay, one is primary structure, secondary structure, tertiary structure, and quaternary structure. So in all these, the final one, quaternary structure, has some uh, uh, they can be visible, and if you dissect them, you will get tertiary structure. Still, if you separate them, you will get secondary structure. Still, if you separate them, you will get straight chain or primary protein structure. Okay, so in primary protein structure, so you will be able to see amino acid sequence, the sequence of amino acids. Okay, so that is about proteins. As I said, most of the drugs, majority of the drugs targets the protein. Therefore, it is very important to understand the structure of proteins, right? So in quaternary structure, what kind of forces stabilize the protein structure? So if you want to understand the forces in between the final protein structure, so you have helical structure in proteins. In between you have hydrogen bonds, hydrogen interactions, and sometimes it has metal ion coordination interactions, like heme. In heme, it is a protein, so it has metal ion. Okay, so the metal ion coordinates with the protein. That kind of interactions takes place, and hydrophobic interactions also exist in protein structure, and also covalent bonds between two sulfurs. Okay, disulfide bond, that interactions also possible. And side chain interaction, hydrogen bonding interactions are possible, and electrostatic interactions. As I said, it has NH3 plus and CO minus. Okay, so in between them, electrostatic interactions also exist. So these kind of interactions, these kind of forces stabilize protein structure. That's how protein exists as quaternary structure. Okay, so it is very important to know and learn about different kinds of interactions. You are already familiar with these interactions, right? You know about hydrophobic interactions, metal ion coordination interactions, and covalent interactions, and electrostatic interactions. Okay, and hydrogen bonding interactions. These kind of interactions, they act as a force to stabilize the protein structure. Okay, that you need to understand. Hmm? So I'll explain you deeply when we come to about proteins and nucleic acids and carbohydrates, lipids. So you are going to study a specific drug action of mechanisms with examples, right? At that moment, I'll explain you very clearly how they bind with the drug, what kind of interactions, chemical interactions takes place. Because of that chemical interaction, what kind of pharmacological or biological effects we can see. Those details you will see later. Okay, and also uh, uh, you already know about proteins and nucleic acids and carbohydrates, lipids. So in proteins and the nucleic acids, if you come to nucleic acids, they have four bases. Okay, those bases you need to you already know the structures of bases: pyrimidine, guanine. Okay, adenine, guanine, pyrimidine, thymine, uracil. Okay, all those structures you should be familiar with them. Okay, so the DNA and RNA composed of those bases as well as carbohydrates. Okay, in the case of DNA, it is deoxyribose, the sugar might exist in that. In the case of RNA, ribose sugar will be exist in that. Okay, so if you think about carbohydrates later you are going to learn about different kinds of carbohydrates like NAM and NAG. NAM, NAM, N acetyl muramic acid. NAM. NAG means N acetyl glucosamine. Okay, they are the sugar moieties. So the bacteria cell wall composed with those sugar moieties. So therefore it is important to understand about carbohydrates chemistry also, so that how drug will interact with carbohydrate structures like NAM and NAG in cell wall structure of bacteria. Okay,